Tantre, and as a co-convener of this event, I'm delighted to welcome such a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward for productive conversation and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in pharmaceutical sector. I'm confident that today's session will uphold its tradition of excellence, providing valuable insights from our respected speaker. Let's seize this chance to gain new knowledge, participate actively, and progress our fields. Welcome everyone once again. I look forward to a productive experience for all the participants. Thank you once again joining us today. So as we gather today to commemorate the 89th SPSR webinar on shelf life assessment of herbal drug products, a new ethical challenge, a distinguished event hosted by Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alok sir, National President SPSR. The society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It's a great privilege to introduce Dr. Gulshan Kumar Bansal sir, professor and former head, Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala, Punjab, as a distinguished speaker for today's webinar. Dr. Gulshan Kumar Bansal sir's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration of shelf life assessment of herbal drug products. We are grateful for his engagement and anticipating an enlightening session. So, before starting, I will go through a guest profile of Dr. Gulshan Kumar Bansal sir. Dr. Gulshan Kumar Bansal sir is a highly respected academician and researcher, currently serving as professor and former head Department of Pharmaceutical Sciences and Drug Research at Punjabi University, Patiala, Punjab. He's also the coordinator of Centralized Admission Cell, chairperson of Institutional Animal Ethical Committee, and president of APTI Punjab State Branch. In recognition of his academic leadership, he has been honored as a CEGR Certified Academic Leader and an APTI Fellow 2024. Dr. Bansal sir holds a b.pharm 1994, m.pharm gold medalist 1998, and PhD. With over 24 years of teaching and 26 years of research experience, he is widely recognized for his contribution in the field of quality control of herbal drug products and impurity profiling. As a mentor, Sir has guided 10 PhD scholars with 5 more currently pursuing, and supervised more than 50 m.pharm dissertations. His scholarly work include over 100 publications in a peer-reviewed journals with a notable H index of 22 and an average impact of 1.8. He is also the recipient of 3 Indian patents and has contributed to book chapters in pharmaceutical sciences. Dr. Bansal sir has successfully completed several research projects funded by CSIR, UGC, and AICTE, amounting to the total grant of over 60 lakh INR. He is a subject expert for academic bodies including NAC, PCI, and other national councils. He also holds life memberships in reputed organizations such as APTI, IPGA, Punjab Academy of Science, CEGR, and SPSTI. Beyond academia, Dr. Bansal sir has made significant contributions, including establishing the alumni network of his department, organizing national numerous national and international conferences, seminars, workshops, actively promoting the rational use of medicine in rural areas, strengthening industry academia collaboration to enhance practical learning, supporting peer network to promote junior senior academic mentorship. So, Dr. Bansal's multifaceted expertise and dedication to advancing pharmaceutical education and research make him a great value leader in the field. It's with a great satisfaction and pride also that we declare we have exceeded 1,300 restaurants globally, showcasing an impressive diversity of countries. Significant attendees is noted from India, Egypt, Ghana, Indonesia, Kenya, Malaysia, Mexico, Nepal, Nigeria, UAE, Uganda, Togo, Sri Lanka. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude goes to each and every person who has taken a moment to participate in this significant event. Thank you for all joining us today. Let's move forward and I would like to welcome our esteemed keynote speaker, Dr. Gulshan Kumar Bansal, sir, who has already joined with us. Welcome, sir.
Thank you, Dr. Junaid, for your kind words. I don't know whether I deserve it or not, but for your very kind and elaborate introduction. Thank you so much. So, should we continue? Should we start with the
Yes,
talk?
please, sir. You can please go ahead, sir. Over to you, sir.
So, please allow me to share the presentation. Yeah. Is it okay now?
Not yet, sir. Not visible yet.
Screen sharing is paused. Resume.
It's visible, sir.
It's okay now? Yeah.
Yes, it's visible.
Okay, so very good morning to one and all here, the organizers of this webinar, Dr. Shashi Alok. Thank you for giving me opportunity to interact with the audience from different parts of the world, and Dr. Junaid for organizing, being the organizer of this particular webinar, and heartfelt thanks to the participants who have joined today. Um, So the topic which I have selected for interacting with is shelf life of herbal drug products, a new ethical challenge. Now, this particular issue about the herbal drug products, it has been, you see, matter of concern for most of the scientists. Now, the point is, um, if we talk about the traditional medicinal practitioners, uh, in India we call them Ayurved. They are very much possessive about, you see, the herbal drug products because yes, there's no doubt that herbal drug products they are very good in treatment of or even cure of chronic diseases. But when it comes to the quality control, when it comes to the shelf life, right, there are conflicting views. The modern science, the regulatory bodies, they need that any medicinal product must have a shelf life. And the Ayurvedic physicians, they contest that the concept of shelf life is not broadly applicable to herbal drug products because they are the natural substances. But we as pharmaceutical scientists who are working with natural products with the modern approach to develop into, you see, modern medicines, we are very much convinced that any drug product which is intended to modify our physiology, which is intended to, uh, uh, you see, cure certain diseases, right? It is through chemicals only, and all chemicals they have a particular shelf life. they have a particular period during which they will remain stable. Beyond that, they will definitely degrade. So if they are degrading, it means their um, efficacy will be affected. Now, the point is how to remove those conflicts, how to elaborate, how to evaluate the shelf life of these herbal products. So these are the few things which we will be deliberating upon during this session. See, herbal drugs, we all know that they are used in our routine life as a health supplements or as a medicinal, and it is used in all parts of the world, be it developed countries, be it developing countries, right? Now there are so many participants from the other parts of the world also, means that it's an international webinar, and you all must be using herbal drugs or herbal drug products or nutraceuticals, herbal nutraceuticals in one or the other form. The acceptability of herbal medicines or herbal drug products, we can see that around more than 11% of the World Health Organization's essential medicine list, they are from the herbal drug products. And more than 80% population of the developing countries, they rely on herbal drug products for the primary treatment of their diseases. Now, the market is flooded with these herbal products because there is a demand. We know that we can sell anything in the name of herbal thing. It is a herbal cosmetic, it is a herbal tea, it is a herbal face mask, it is a herbal shampoo. You talk anything, right? In the name of the herbal, you can sell anything. So the market is flooded with an artillery of these products from the different manufacturers. And the matter of concern is that these herbal drug products, right, although they are required to be manufactured in a pharmaceutical manufacturing unit as per the GMP guidelines, which is proposed by WHO guidelines, by the European Medicinal Agency also. But given to some flexibility in some countries, uh, many of the manufacturing units, they are operating from 2 rooms or 3 rooms or 4 rooms units, where there is no proper quality control, there is no proper storage, there is a forget about the accelerator stability studies, forget about the long-term stability studies, forget about the toxicity studies. Even the manufacturing facilities, they are not, controlled. They are not hygienic. Right now, in this cases, the issue becomes how we can ensure the quality, safety, and efficacy of such products. Because those products, many of those products, they are not manufactured as per the GMP guidelines. Although the guidelines are there in place, but how stringently they are enforced, how stringently they are made to follow by the manufacturers, that remains questionable. So, to ensure quality, safety, and efficacy, that becomes a primary concern for a herbal product. Now, US FDA, they have laid down a very stringent criteria uh, for for approval of any herbal drug product which is having a specific therapeutic benefit. Means if we say that this particular product, for example, Sagizium cumini, which is in India we call it a jamun, right? That is a fruit. So if we using that particular product for the treatment of diabetes, right, then it has to be approved as a medicinal product by the US FDA. Only then it can be promoted as anti-diabetic. Otherwise, it can be promoted only as a health supplement. Whereas the European Medicine Agency, they require identification and standardization of a herbal medicinal products in terms of therapeutic or analytical markers. So they are not treating those herbal medicinal products as solely medicinal products, which is very stringent as per the US FDA in terms of US FDA. So European Medicine Agency is quite lenient in that way. Of course, they need stability testing of the herbal products. So there are some stringent criteria by the European Medicine Agency, but US FDA is the most stringent one. So this is the different guidelines in the different parts of the world. Every country has their own guidelines. In addition to this, there are some common guidelines like US FDA, ICH guidelines are there, European Medicine Agency, EMA guidelines are there. You have Brazil, you have you see Australia, you have China, you have Russia, you have Canada, you have India, right? So our UAE is there. So almost every country they have their own guidelines for approval of herbal products as nutraceuticals or as medicinal products in their country. So this quality, safety, and efficacy, if they are maintained as per these individual national guidelines, then those products are said to be approved for therapeutic purposes or for nutritional purposes. Now, the point is how to establish quality and safety and efficacy. Now, the quality, if we talk about, it is in four ways. One is physicochemical testing, that is both qualitative and quantitative. Then we have a microbial contamination, pesticides and heavy metals, adulteration and substitution. So these four parameters, these four broad parameters, they come under the quality. And for safety and efficacy, we can rely upon literature reports or we can experimentally establish. Literature reports, for example, we know that turmeric is commonly used as anti-inflammatory as well as antimicrobial. Now, it has been into use since ages. Now, there are literature reports based upon which its patent claim by United States of America was nullified. Uh, so, if there are extensive literature reports available on safety and efficacy of that particular herbal product or herbal raw material, then we don't need any experimentation for that. It can be considered as safe and efficacious. But if we are mixing the two, you see, products, for example, if we talk about turmeric, if we talk about, let's say, Azadirachta indica, right? Both are the different herbs, both are having their different uses, individual uses. But if we mix the two, right, that will be treated as a new product, right? Now, in such cases, the safety and efficacy has to be done experimentally, right? So these are the issues which we have to consider while evaluating quality, safety, and efficacy. Now, quality, establishing quality of a herbal medicinal product, that is the most challenging task. We will come to the point why it is the most challenging. No, but it is important to establish quality because establishing quality will ensure safety, establishes it's a batch to batch consistency, and it also ensures therapeutic effectiveness. When you say about quality, safety, and batch to batch consistency, for example, uh, we are having a product of, let's say, Vidania somnifera. Now, Vidania somnifera may be obtained from some source in which the Vidania somnifera is cultivated in farms under controlled conditions using good agricultural practices. But Vidania somnifera is also widely grown along the roadsides. Now, from what is the source of that raw material? That is important. because because of the variation in the geographic locations, the climatic change, their secondary metabolite composition will be different, their phytochemical profile will be different, and of course the content of the heavy metals will be also different in those herbs. So batch to batch consistency and safety, that is very important because of the variability in the collection of the raw materials in the herbal products. Now, if you see the quality control strategies, what are those? Like authentication and quality evaluation. So first part is authentication. We need to authenticate the product which we are, the herbal product, which herbal raw material which we are using, whether it is a clear, whether it is a actual or not. For example, uh, Becopameneri and Centella asiatica. Both are commonly known as in Indian literature as Brahmi, right? But both are botanically different. Their morphological characters are different. Their phytochemical profiles are different. So, what exactly it is, whether it is a Centella asiatica, whether it is a Becopameneri, that we have to authenticate with the help of with the help of a botanical expert using macroscopy and microscopy. Then quality evaluation and sanitation, we have to see physical evaluations, biological evaluations, and chemical evaluations. In the biological evaluations, toxicological studies, microbiological studies, and pharmacological studies, they are the part of that. And in chemical evaluation, we will do the quantitative, qualitative, heavy metal analysis, pesticides, markers, secondary metabolites, DNA, or so on. So, this quality, safety, and efficacy, if we want to establish, it is cannot be just physical or chemical or biological. It is combination of all three evaluations. We have to evaluate the physical characteristics, physical quality, chemical quality, and biological quality. Only then we can ensure quality, safety, and efficacy. Because the purpose of anything which we consume is to get the intended purpose. For example, if we are consuming, let's say, a turmeric tablets, it means we need anti-inflammatory action. If we are applying topically on a wound, we need anti-microbial action. Now, if even if it is qualitatively okay, means let's say it contains curcuminoids or curcumins as per the norms, but if it is not eliciting the response, then it is of no use, right? So, quality, safety, and efficacy means physical, chemical, biological evaluations. All three are required for establishing quality, safety, and efficacy. Now, does we have established the now quality using physical, chemical, and biological modes. Now, does this simple quality assessment of a herbal drug or product ensures that it will remain consistently therapeutically active throughout its shelf life? Because as per the modern guidelines, we have to mention a expiry date on any product. Now, whether it is a nutraceutical, whether it is a pharmaceutical, whether it is a medicinal product, whether it is a surgical product, on every product there is a written best before use or expiry date. So that we call it a shelf life. Now, whether that will remain active through the shelf life. Now, this is where most of the Ayurvedic practitioners they contest that herbal drugs are natural, so they don't need any shelf life. They remain stable. They are traditionally considered safe. Therefore, there is no need to carry out any, you see, safety and efficacy studies. There is no need to carry out any shelf life assessment studies. And they also contest that many herbal drugs they are stored with the traditional medicinal practitioners for long periods of time, maybe 5 years, 10 years, 15 years, 20 years, because there are some herbal products which are collected from far-flung areas, from very you see high altitude areas, right, or from very you see remote areas. So it's not possible to collect those raw materials uh, in routine, right? For example, if you talk about rhubarb, now rhubarb is available in the high altitude, in the higher altitudes of Srinagar, right? Now, it is not possible to collect this rhubarb throughout the year, means every year, right? Either it has to be cultivated or it has to be collected. So they remain with those protections for many periods of many long periods of the time, right? So they say that they don't need any shelf life. So, if we believe that practitioners, because they are the Ayurvedic, you see, they are the herbal scientists, does it mean that the herbal drugs do not need any shelf life? Now, from the traditional practitioner point of view, I can say, because I'm possessive about that, I can say yes, they don't need any shelf life. But from a pharmaceutical scientist point of view, I must contest that every herbal product or every medicinal product or every nutraceutical product that needs a shelf life. So, what is shelf life? We all know about this, just a very brief. The time period during which a product, medicinal product or a food product, retains its physical and chemical integrity and remains suitable for its intended purpose. That's important. That's why I have specified the definition of shelf life. Otherwise, we all know what is a shelf life. It must remain suitable for its intended purpose. It's a physical or chemical integrity may remain same, but if it does not remain suitable for the intended purpose, it is a violation of the shelf life. If it remains suitable, but its physical and chemical integrity change, then again it violates the shelf life norms. So all the three, physical, chemical, and biological characteristics, they have to remain within the set limits. World Health Organization, they define the expiry date or the shelf life as a period of the time during which an API, the active pharmaceutical ingredients or a finished pharmaceutical product, if stored correctly, is expected to comply with the specification as determined by stability studies on a number of batches of the API or FPP. The shelf life is used to establish the expiry date of each batch. Now it has to be done on each batch before it is released by the pharmaceutical industry. Now, this is a WHO definition for any API or pharmaceutical product. Now, why there is a need of shelf life assignment for herbal drugs? Because we are considering that they are the shelf. They are the, you see, natural one. Now, if we compare the two, synthetic drug products and the herbal drug products, they are similar to some extent, they are different to some extent. What is the similarity? Both are having therapeutic or nutritional values we consume for these purposes, and both have to be manufactured as per the GMP guidelines, good manufacturing practices guidelines. But there are some differences, contrasting differences. What is that? The synthetic drug product, for example, we are talking about any allopathic medicine, for example, crocin, for example, dolo, for example, any moxicillin tablets or moxicillin capsules. So they contain single or multiple known drugs. We know that it contains these particular active ingredients. But in case of herbal drug products, we just know that it contains this particular herb or that particular herb. It can be monoherbal or it can be polyherbal. It can be monoherbal, biherbal, triherbal, tetraherbal, pentaherbal, polyherbal. It may be containing 20 herbs, right? There are variety of the polyherbal formulations products available. So the let's say if we talk about just one herb, does it contain only one chemical? No. One herb contains 10, no, if not hundreds, right? More than 20 or 30 chemicals. And every chemical is behaving differently. And on the top of it, we do not know the complete chemistry of those herbal constituents. So in case of synthetic products, the chemical nature is known. Let's say in crocin, we know that it contains paracetamol. So what is paracetamol? We know that it is a one phenolic group is there. There is a one acetamido group is there, right? And what is acetamido? They have the particular stability issues, right? It can undergo degradation, right? We know the chemical structures. But in case of herbal products, we know the structure of only a few phytoconstituents which are characterized by some other scientists using the for using some sophisticated techniques. Like if we talk about the turmeric again, right? We know that it contains curcumin or curcuminoids. What else? There are so many other chemicals which are available, right? If we talk about, let's say, uh, Azadirachta indica, right? It contains azadirachtin, right? What are the others? Cinnamons, they contain this quinine, right? Quinine, quinidine, cinconidine. They are known. The four chemicals are known. But there are so many others which we do not know. And what is their chemical stability? How, what will be going to their, you see, therapeutic or nutritional or toxic effects? We do not know, right? So all these are the two contrasting difference. It means that almost every chemical which we are consuming, it is liable to undergo change or degrade under the influence of environmental conditions. That is a humidity, temperature, light, air, right? And if they are undergoing these conditions, then their composition is going to change. Now, see here, these are the different possible interactions between the different groups of plant constituents. Now, in dispensing pharmacy, in first year of the b.pharm program, we uh, learn about the incompatibilities. So see here, what are the different types of reactions that are possible in the different types of phytoconstituents, like heavy metal chlorides, they can interact with the tannins, they can interact with proteins, glycosides, they can interact with the heavy metal chlorides, they can interact with the tannins, they can interact with the organic plant acids, proteins with the this tannins and this ethanol, polysaccharides, with the alkaloids, with the tannins, with organic acids, with ethanol, or so on. So there are the different, you see, possibilities of the interactions. So in a herbal product, which is containing hundreds of chemicals belonging to different chemical classes, and they are interacting with each other. So see the complexity of the chemical complexity of the herbal product. So we are not able to actually identify all the constituents. Forget about the all the constituents. We are not able to identify the major phytoconstituents. So the polyphenols, they form reversible complexes with proteins and polysaccharides by hydrogen bonding and hydrophobic interactions. Water solubility of tannin is increased in the presence of water-soluble glycosides. Alkaloids are precipitated in terms of tannins. Polysaccharides, tannins, and lignins, they form mono or bidentate complexes with heavy metal ions. So let's say if the two kind of constituents, for example, uh, we talk about the polysaccharides and heavy metals. Let's say these two are not present in one herb, but they are present in two different herbs. And when the two herbs are mixed together to formulate a bi-herbal product, then those polysaccharides and heavy metals, they will not be available for absorption because they are forming the complex. So that's why the efficacy is important when we are using a mixture of herbal products. This is the kind of you see chemical example of chemical transformation which occurs in herbs, isoliquidity which undergoes conversion to liquidity. Now, this isoliquidity, it is a very potent GABA A benzodiazepine receptor uh, um, compound. It is a positive allosteric modulator, and it is a 65 times more active than diazepine. But in acidic environment, it undergoes rearrangement, intramolecular reaction to form liquidity which is very, very less active. So the biological activity is reduced. Now, rutoside, we all know that quercetin. If we want to do the chemical analysis or chemical standardization of any plant which is containing flavonoids, we determine total flavonoidal content in terms of quercetin, right? But the main active constituent is rutoside. The rutoside, if you see quercetin with the one glycoside unit, that is a quercetin, and quercetin with another glycosidic unit, that is a rutoside. Now, if uh, this rutoside, it undergoes glycosidic hydrolysis, it is converted to quercetin, then it is not as effective as rutoside, which is anti-fatigue, anti-inflammatory, anti-diabetic, and antioxidant. Another example of artemisinin, which undergoes degradation under different chemical environments. Then menthol, which is undergoing photochemical reactions, which is a natural pesticide. So if it is undergoing a photochemical reaction, it will be no more a pesticide. And these are the different phenolic compounds like flavonoids, flavonols, caffeic acid, or so on. They undergo, you see, this reaction with the amino acids, right? So this phenols, they are converted to true oxidation quinones, and this quinones will undergo reaction with the amino acids or the other amine products to give the form of coupling products. This is, you see, this reaction is mainly responsible for browning of the plant material. We know that banana, it undergoes a browning. We know that any vegetable or any fruit, they undergo browning with the storage. That browning is because of this reaction, the Schiff reaction. It means if the browning is taking place, the phenolic components, the flavonoids, which are antioxidant, they are no more available, or they are less available. So these intramolecular or intermolecular reactions, they under the influence of different chemical environments during storage, right? Of course, they will change the chemical profile of the herb, right? So if the chemical profile has changed, it means their therapeutic effect, their nutritional effect, their toxic effects, their safety profile, that will change. So this is a sufficient scientific basis for the need of shelf life assessment, which we contest to the traditional medicinal practitioners. Now, how we determine the shelf life through stability studies. So that stability testing, it provides evidence on how the drug substance or drug product quality varies with the time under environmental conditions during distribution and storage. So different guidelines across the globe, WHO guidelines, EMEA guidelines, Indian guidelines, they have given, you see, specific instructions like WHO guidelines for GMP of herbal medicines. Stability data are always required to support the shelf life purpose for the finished herbal product. Very explicitly, they have cleared. The stability of preservatives and stabilizers should be performed, right? So for the excipients also, they need, because they are the synthetic molecules, not the neutral one. Then this is another WHO guidelines for methodologies on research and evaluation of traditional medicines. It also says that the physical and chemical stability of the product should be tested under storage conditions, and the shelf life should be established by the systematic stability testing. European Medicine Agency, they have their own guidelines. Committee on Herbal Medicinal Products, HMPC, or a committee for medicinal products for human use, committee for medicinal products for veterinary use. For all these guidelines, they have given the guidelines on quality of combination herbal medicinal products. And these guidelines, which are passed in 2008 and still applicable, they say that under the title Stability of Herbal Medicinal Products. The stability of the combination of herbal medicinal products should be determined in accordance with the existing guidance on stability and the specific herbal guidelines. So there are the regulatory basis. So in different parts of the world, as we have also seen in the previous slide. In Canada, we have the Natural Health Product Directorate. In South America, we have the Ibero-American Network on Phytochemical Products. In South Africa, we have the Medicinal Control Council under Medicine Act. In the UAE, we have the Gulf Central Committee for the Drug Registration, EMA in the Europe. In India, we have the Drug and Cosmetics Amendments Rules, Asian guidelines, therapeutic guidelines, right? So these are the different guidelines across the world, which specify that stability testing has to be done. Now, this shelf life concept, which we are discussing or which we all know, it is not new. It is not new to the Indian culture also, right? In India, in the traditional literature, we call this shelf life as Savirtya Avadhi. Now, this is a Hindi word, the Sanskrit word, right? The international participants may be having a little difficulty in understanding this, but this is, you see, the legacy of the Indian system of medicine, because they have established the concept of herbal medicine thousands of years ago. They call it Savirtya Avadhi, means the time period during which the virya, the again Sanskrit word, the potency, which means potency of any drug remains unaffected due to environmental or microbial deterioration. So a drug that may be old or fresh can be utilized for therapeutic purpose, right? Which up to a period which its qualities and remains in intact conditions. So that is mentioned in our Veda, which is a Shushruta Samhita. Now, Savirtha Vedi or the shelf life, if you see the for the many Ayurvedic drug formulations, it was proposed by different scholars in the 12th AD. In 12th AD, right? So that is a pretty long time back, more than 800 years ago, 900 years ago. Like decoction, they have a shelf life of 3 hours. Paste, they have a shelf life of 3 hours. Expressed juice, they have the life of 3 hours. Then you have a churn, right? The powder we call it, 2 months or 3 months. Then pills, that's the tablets, 12 months. So they have these different kind of products, formulations. They have their specified shelf life in our traditional literature. Now, based upon that literature, drugs and cosmetics rules have been amended with first amendment in 2005, then the second amendment in 2014, uh, in which they say that the herbal drugs or the herbal medicinal products, they must carry some expiry date. So see here, the date of expiry of Ayurveda, Siddha and Unani medicine shall be conspicuously displayed on the label of the container or package of an Ayurvedic, Siddha and Unani drug, after which they shall not be in circulation. Shelf life for this medicine shall be as follows. It means to up to 2005. Up to 2005, there was no shelf life written on any herbal product. It was mentioned only date of manufacturing. There was no date of expiry in the product up to 2005. It is only after 2005 that the concept of shelf life has come to existence. So according to that rules or the amendments, see the powders, I just read it, translate it to the English, like churn, the powder, 1 year, tablets, 2 years, right? Then we have the rasaushadhis or asavrishta, they have do not have any expiry date. Now this is a matter of concern. The first matter of concern is there are certain products in which they do not have any expiry date. Rasaushadhis, asavrishta, then this perpati or so on. How is it possible that the product is not having any shelf life? Scientifically, it's not possible, right? Then the second issue, which is worth discussing here, is that the guidelines say that the powder must have a shelf life of 1 year. Now, which powder? Whether it is a powder of turmeric, whether that is a powder of, curcuma longa, whether that is a powder of, let's say, Zadica indica, whether that is a powder of Ocimum sanctum, which herbal product we are talking about? Because every herbal product, every herb, not product, every herb has its own phytochemical composition, means the chemical profile is different. If the chemical profile is different, their chemical stability is different. So, if we are saying that Ocimum sanctum powder is stable for 1 year, we cannot extrapolate that 1 year to the powder of any other herbal raw material. So, this is the catch. So, this is the other one, right? Like say here, Suvarna, Rajata, Loha, Mandura, Abhark Bhasam, right? Shashanka Bhasam, they do not have an expiry date. Bhasam, they do not have an expiry date. Now, it means, although there are some guidelines, if we talk about the international guidelines, WHO, EMEA, they have certain guidelines which say that the shelf life has to be established on the basis of stability testing. But in Indian context, they say that the shelf life can be given as per the amendments, and which is scientifically not valid. Now, what are the different steps in the stability testing? If we want to perform the stability testing, there are some protocols. So we have to first lay down a properly designed test protocol, then conduct the stability as per the protocol, and then establish or predict the shelf life on the basis of that data which is generated after the analysis of stability samples. So these are the different guidelines. This is different conditions which uh, we use for the for evaluating the stability, long-term stability studies, intermediate stability studies, or accelerated stability studies, depending on the different parts of the world, long-term stability studies are used. Then we have the different guidelines like EMEA, we have the different guidelines. For WHO, we have the different guidelines. And now, these are the protocols for the different herbal drugs. So, a herbal substance in its entirety is regarded as active substance. This is what we are contesting since the beginning, because it is containing multitude of constituents, and every constituent has its own biological activity, right? So, herbal drug as such is known as active substance. Then polyherbal medicinal products. So for this active substance, we have to carry out the stability. We can use a fingerprinting chromatogram to establish their stability. Because from the fingerprint, we come to know which particular phytoconstituent is decreasing or increasing, or which particular new interaction product. Interaction product means interaction between the two phytoconstituents. It is rising. In case of polyherbal medicinal products, the stability of the product will be determined by appropriate fingerprint chromatograms. Again, fingerprints. Then in herbal medicinal product with known therapeutic phytoconstituents. If we know that, for example, turmeric, we know that curcumin is the active constituent, then we can standardize it with the respect to curcumin, and and its content should not vary by plus minus 5% till its shelf life. If we have some analytical marker, means in which the herbal medicinal product, which is with the no known therapeutic constituent, we don't know the therapeutic component, but we know some components which are used as analytical markers. then their content can be vary by 10%. Now, the various factors which affect the shelf life, that is a physical, chemical, and microbiological, and these factors are not actually they exist in isolation, they are related to each other. Any change in the chemical uh, nature because of oxidation, hydrolysis, crystallization, or so on, that will lead to change in the physical nature, that will lead to change in the microbiological. So they are related to each other. So we have to evaluate a herbal product during its shelf life assessment in terms of physical nature, chemical nature, and microbiological nature. And if we see the current reports which are available in literature on the what kind of studies are performed for shelf life for stability testing of herbal products, 67% means most of the studies they have done only physicochemical analysis or chemical analysis. 14% they have done physiobiological or only biological studies, means they have uh, performed the stability studies, got the stability samples, and they evaluated their chemical profile, chemical, or their biological study, means they have determined their biological efficacy, which sound justified because ultimate aim is to see whether the product remains effective or nutritious during its shelf life. 11% they have used just physical evaluations. Let's say they have carried out studies on emulsions. So what the physical studies we do on the emulsions, like whether there is emulsion breaking, whether there is a change in pH, change in color, or so on. And it is of no use if we say uh, whether it does not reflect whether the chemical change has occurred, if it has occurred, how much it has occurred, right? Whether the biological activity remains the same or not, it's not going to do any, you see, not to provide any information. And there are only 8% studies which have performed all three evaluations: physical, chemical, and biological. And the second aspect which I want to bring in your notice is that in these studies, there is a wide variability in the conditions which are employed for stability evaluations. See the temperature. The stability study is performed at a temperature from -80 to 100 degrees Celsius. Now, I do not understand what is the rationale of carrying out study at -80 degrees Celsius. Are we going to sell that product in Antarctica? Then -80 we can consider whether it will remain stable. And as a general rule, we know that lower is the temperature, more stable it will be, right? We can increase the temperature for stability testing, but why to decrease? So there are some literature reports which are reporting this. I don't understand what is the rationale of that, right? 100 degrees Celsius, what is the rationale of carrying out stability study at 100 degree? Stress degradation studies or degradation studies that are understood. So those are degradation studies, not stability studies. Relative humidity is varying from 0 to 100 degrees. Duration few hours to 3 years. 3 years is okay. Few hours stability, it's no use. So there is a wide variability, means these conditions which are used by the different researchers across the globe to perform the stability studies on herbal products, they are just selected without any rationale. Now, what third aspect is what challenges we face when we carry out the stability testing of herbal products. The first challenge is the chemical complexity. Second is the biochemical composition variability in the raw material. Then substitution adulteration, and fourth is the selection of markers. The chemical complexity, as we know that, uh, as we have already discussed, that a single herbal product, it contains numerous chemicals. So the chemical complexity is very high, even in a single herb. And if we use multiple herbs, then that chemical complexity multiplies exponentially, right? And we are not able to identify the precise therapeutic marker, right? So if we know the therapeutic marker, then it is okay. That is a good for us. We are lucky one. But if we do not know, then we have to develop a comprehensive chromatographic fingerprint so that we have a complete list of chemicals which are present on those in that particular product. Second challenge is the variability in biochemical composition. And this is because of the changes in the agriculture practices, because of the change in the climate conditions, because of the change in the, you see, latitude, longitude, right? Let's say the turmeric, which is grown in northern part of India, it is a much different from that which is grown in southern part of India. The Indian, you see, turmeric is different from the, you see, African turmeric because of the, again the different geoclimatic changes, right? So biochemical composition is variable. Now, when we are developing a raw material into finished product, the source of the raw material, that is important. We must know what is the source, whether it is the same source which we have used. Let's say we have developed a liquid oral product of any herbal of a plant. Now we have fixed its dose as, let's say, 10 ml based upon its standardization. Now, when the next batch is manufactured, if its raw material is from the different source, then we have to affect the dose. Either we have to change the dose or we have to change the composition or change the concentration of the herb in the product. So those modulation has to be done. Substitution and adulteration. That is a very serious concern in herbal products. For example, convolvulus pluricolis. In India, we call it uh, this shank pushpi, right? Now, this is having four variables. Convolvulus pluricolis, convolvulus decussata, alicinoid, this alicinoids, and then convolvulus ternatia. Now, all four traditionally are called as Shankpushpi, but they are species-wise different or genus-wise different. It means their phytochemical profile is also going to change, right? Now, which Convolvulus pluricolis, which Shankpushpi which we are using, which you see Convolvulus species is supplied to the us to manufacture that particular product, that has to be. Now, out of this, the Convolvulus pluricolis, that is the most active. That is the most um, rich in the therapeutic markers, right? Then, if it is uh, substituted with the other things, then that is a decreation in the activity. Then Bacopa monnieri and Centella acetica, Bacopa monnieri is expensive one, Centella acetica is a cheaper one, so it is a commonly substituted, right? And both are commonly known as a Brahmi, as we have already discussed. Then the selection of markers. Which marker we have to select for standardization? Therapeutic or analytical? If we have a therapeutic known, then we can assign. But let's say, if we take the case of a turmeric, we say curcumin is there. Is the curcumin only anti-inflammatory or antimicrobial? No, they are the other constituent also, right? So one thing is the selection of the therapeutic marker is limited, or in some cases, there may not be any therapeutic marker known. We may have just analytical markers. So which marker is to be selected? That is another challenge in the stability studies. For example, with anolites, we know that they are the active constituents, so we use the markers for Vidania somnifera. Then riboside or stereosides, they are the natural sweeteners. So they are used for standardization of stevia rebudiana, right? Then curcumin for curcuma longa. Then this hyperforin is used for antidepressant activity of hypericum perforatum. So Whenever there is a known, we can use it. But in most of the cases, the therapeutic marker is not known. And if we see the reports which are available in literature, what stability conditions they have used uh, to evaluate the stability studies, see 76% of the studies which are reported in the literature, they have used they have used stability conditions, means accelerated or this long-term stability studies, their protocols, their duration, right? They are not at all related to the recommended guidelines, like accelerated at 40 degrees Celsius, 75% RH, intermediate at 35 degrees Celsius, 65% RH, long-term or so on, right? They are not obeying any rules. Then 23% studies, they are complying with the storage conditions, right? And sampling schedule recommended by accelerator stability studies. Accelerator stability studies are okay in 23. But there is only just 1% studies which are reported in literature which comply with the conditions for long-term as well as accelerator stability studies. So we have gathered the information around for around 200 research papers. And what we came to know that out of 200, only three papers uh, which were showing, which were using the stability study conditions and the sampling protocol strictly as per the ICH or WHO guidelines. Others, they were not following. It means, although the guidelines are there, but we are not following for establishment of stability studies. Now here I will give you a case study which we did in our laboratory. Um, we have carried out stability studies on this marketed product. In India, we call it a shank push pea syrup, convolvulus pluricolis, which we have just talked about. Now, according to that rules, which are mentioned in the Drugs and Cosmetics Amendments from 2014 or 5, um, claimed shelf life is 3 years. So we did its long-term stability conditions. We placed it for three on the in the long-term stability conditions, means at 25 degrees Celsius and 80%, 65% RH. And we carried out the physical, chemical, and biological evaluations. And in the physical evaluation, because it is a syrup, so we have carried out its um, change in the color, taste, odor, viscosity, pH, and all that. And we have seen that in the long-term conditions, their viscosity has slightly decreased over the period of time. Then if we perform the chemical evaluation performed in different ways. One was the percent change in the gram equivalents of gallic acid, which we generally do to evaluate for while evaluating antioxidant activity or total phenolic content. So see the total phenolic content. We have, and as per the guidelines, we have conducted these studies on three batches, A, B, and C. So the total phenolic content was decreased from 14 to 25% within just 1 year under long-term stability conditions, not under accelerated conditions, right? So there is a decrease in the total phenolic content in the formulation. Scopolamine was taken as a therapeutic marker because that is a therapeutic marker for convolvulus pluricolis. And we have developed a HPLC method for that. And these are the some you see components, excipients which are used in the product, right? And if we see the scopolamine content in all the three batches, the scopolamine content was reduced by 56%, 59%, and 31%. Means almost half of the content was degraded within just 1 year. It means the convolvulus pluricolis has degraded to something else. Means its scopolamine has degraded. It means it has undergone some chemical change. So the original form of the convolvulus pluricolis has changed. Then we also did the biological evaluation. Now, what is the intended purpose of this convolvulus pluricolis? it is used to enhance memory. And this SIRP is used not only just to the young ones, but to the children also, so that their memory is boosted. It is also given to the geriatric patients so that they can, their declining memory can be, controlled. So we have performed biological evaluations in two types of biological evaluations. One is anti-radical activity, we call it antioxidant, and ACH inhibitory activity. Both are in vitro. ACH inhibitory activity is done as a measure of memory activity, right? So if we see that mm, anti-radical activity, it has decreased from almost 65% to 50%, and ACH inhibitory activity has also reduced from 38 to 30, this are on an average of 25. So there is a decrease in ACH inhibitory activity, there is a decrease in anti-radical activity. We also did this in vivo activity also. That data is not shown here. Uh, we have used this Morris water maze test, which is a learning model, learning acquisition cognition model, and there was also decrease in the learning ability of this memory activity with the phyto with the stability samples. So what we concluded from the data that although the claimed shelf life was 3 years, but the actual shelf life was less than 1 year because their scoporating content has reduced, their ACH inhibitory activity has reduced, their antioxidant activity has reduced, total phenolic content has reduced, and in vivo memory enhancing activity is also reduced. So see, according to those guidelines, it must contain 3 years, and but its activity is reduced, right, to 50% in just 1 year. It means it is not of use even after 1 year. And these products which we have tested, these are not manufactured by the local manufacturers. These are manufactured by the multinational companies, which are very well known in India. Because of the proprietary issues, we cannot take their name. We can disclose their name. That's not ethical here. But this is the practice which is being followed all over the industry involved in herbal drug products. So, what we need to understand, there is a need for mandatory evaluation of physical and chemical stability along with the major biological activities of a herbal drug product during systematic stability studies. Now, although the WHO guidelines, EMEA guidelines, they say that you have to perform the stability studies and you have to perform physicochemical, but I suggest biological studies also have to be done. They must be made a part of this stability testing protocol for herbal products. Therefore, the guidelines have to be amended, and the therapeutic activity evaluation has to be made integral part of the stability testing protocol. So, from this presentation, from this learnings, what I suggest a take-home message: herbal drugs are good one, but we have to use them with wisdom and caution. With wisdom and caution. Like this is one example which I just disclosed. Now, what we can do as a customer, what we can do as a person, if we go to purchase any herbal product from a medical store, we have to check its expiry. If its expiry is written, let's say for 3 years, right? Then don't buy it if it is older than 1 year. This is what I recommend. This is what I follow, right? Let's say we went to buy a product of, let's say, herbal tablets, right? And its date of manufacturing is, let's say, December 2023, and expiry is, let's say, November 2026, 3 years, for example. December 2023 to 1 year, November 24. So I assume that it is active up to November 2024. Right? So I should not buy this. The medical store, the pharmacist, the chemist, he may protest, he may contest that its expiry is still way away. But we can say no, we want a fresh one. If they have the fresh one, buy it. If they don't have fresh one, don't buy it. Ultimately, they have to send it to the manufacturer back. This is only we can use it judiciously. Thank you. Thank you so much for your patience sharing. So if there is any query or any doubt or anything new which you can add to this presentation, you are most welcome.
Thank you. Thank you so much, Dr. Gulshan Bansal sir, for delivering such an insightful and thought-provoking lecture on shelf life assessment of herbal drug products and a new ethical challenge. You have given us a deep experience regarding the complexities of stability, whether you have talked about the evaluation parameters also, regulatory gaps also, ethical concerns in the herbal formulations, and we have garnered a very good understanding about it, sir. It was a thought-provoking, especially the case studies you mentioned. We sincerely appreciate the time and knowledge that you have shared with us today, sir. Uh, Definitely, your session has sparked a meaningful reflection of scientific and ethical dimensions of herbal drug development. Thank you once again, sir. So we have received few questions. Under which I have highlighted two, three. So the first question will be, are there established guidelines from regulatory bodies like Ayush, WHO, or ICH for shelf life evaluation of herbal products in India?
Uh, ICH guidelines, WHO, they have some guidelines which are applicable to whole of the world. And they have specifically said that the stability studies on herbal products also has to be done as per the protocol. And what is the protocol? Accelerated stability studies, intermediate studies, and long-term stability studies for 6 months, 1 year, and you see up to the expiry date. Now the issue is what needs to be evaluated. In case of synthetic herbal products, we know that it contains this particular 2 or 3 or 4 chemicals. We can monitor their content, and if their content decreases, we know that its activity is going to decrease. But same thing we cannot extrapolate in herbal products. Even if we know one therapeutic marker, it does not mean that its biological activity will be totally abolished or it will decrease. And we have seen the example of, you see, Drugs and Cosmetics Amendment rules in 2014, which is applicable in India. In India, there is rules for setting the shelf life. What are those rules? They say that you fix 1 year of shelf life for all powders, whether that powder is of ashwagandha, whether that powder is of turmeric, whether that is of you see black pepper, whether that powder is of let's say osimum sanctum, whatever. How the two different set of chemicals can have a same shelf life? Not possible. So technically they are wrong. So I will say
Right, sir.
technically there are no such guidelines in place.
One more question we have received, sir. How can impurity profiling aid in defining expiration dates for herbal products?
No, the concept of impurity profiling is not truly applicable to herbal products. Because what is an impurity? Anything other than the active ingredient. If you see the simplest case of paracetamol, its impurity is paraminophenol. So if paraminophenol content is increasing, it means the paracetamol is decreasing. But in
Okay.
case of herbal products, there are hundreds of chemicals. And there is a possibility of intermolecular reactions. So there will be interaction products. We are not able to isolate all therapeutic active markers. How we can isolate the impurities? How we can isolate, how we can identify the interaction products? So what we can do? So in as far as my knowledge is concerned, I don't think that the impurity profiling can be applicable to herbal products. What we can apply to the herbal product is, let's get a comprehensive chromatogram fingerprint of a standard drug and then generate stability samples and see what changes are taking place in those fingerprints, right? And let's say try to identify through the sophisticated techniques like LCMS, LCNMR, because we cannot isolate those products in sufficient quantity, right? So only using sophisticated techniques, we can identify their structures so that we can come to know what kind of intermolecular reactions are taking place, and we can predict, you see, postulate uh, uh, appropriate stability condition, storage conditions.
Right, sir. We have a last question that, sir, it's a very interesting one. Can nanotechnology or modern formulation techniques enhance the shelf life of herbal products?
Yes, definitely. Nanotechnology, nanobiotechnology, that is, you see, that has been a talk of the town, and now everyone is using it. Their stability can be increased, but the my major concern is how to do the stability studies, how to establish the shelf life. Means if we want to establish the shelf life, right, what parameters have to be evaluated? That is a
Understood.
point
Right.
of concern.
Thank you so much once again, sir, for answering all the questions so wonderfully and with such a clarity. Your insightful and detailed responses have added a great value to our discussion and helped deepen our understanding on the scientific and ethical dimensions of shelf life assessment in herbal products in India. So we are concluding this ceremony of 89th SPSR International Webinar. We extend our heartfelt thanks to Dr. Gulshan Kumar Bansal, sir. It was an honor, sir, for delivering a highly insightful and thought-provoking session. Today's session brought to the light to the critical importance of scientific validations, regulatory alignment, ethical responsibilities in the context of herbal drug product stability. Dr. Bansal sir's expertise helped us to better understanding the complexities of herbal productions, formulations, and shelf life assessment challenges. Thank you so much, sir. I would, on the behalf of SPSR, we sincerely thank you for giving your valuable contribution. And thank you once again, sir. And I would also like to thank our national president, Shashi Alok, sir, for honoring us with his presence. I would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, ma'am. Due to some work, she was not able to attend. But without her expertise, it's impossible to complete this webinar. We will look forward for participation in future SPSR endeavors also. Thank you so much, sir. Have a wonderful day. Thank you. It was an honor listening to you, sir.
Thank you, Dr. All the best wishes.
Thank you, sir.